Well, hello, everyone, and welcome to today's webinar focusing on gene edited crops, where our speaker is particularly going to focus on experiences in product development and global regulations. I'm Chris Boomsma, CEO here at the Council for Agricultural Science and Technology. It's our privilege today here at CAST to team up with the Society for In Vitro Biology to bring you this webinar. Before we get started, just a few words about CAST. First of all, it's an organization over 50 years old that's provided credible, unbiased, science-based information for those years, our principal audience being policymakers, the media, the private sector, and the public. Next slide, please. We are a US-based organization, although we are internationally connected, and we are membership based, both organization level and individual level. And we'd love to have you or your organization join us. We do span all of the agricultural science and technology arena from animal to food to plant, and we are nonpartisan, science focused, and all of the items that we produce, that is the educational products, are free of charge. Next slide, please. Here's our mission, which I mentioned earlier, and I mentioned that one of our chief um, strengths that we have is to convene and coordinate networks of experts. We have many subject matter experts that contribute to the programs that we have and the information that we put out. Next slide, please. Speaking of experts, here's the makeup you can see from academia, nonprofits, government, companies. They come from all those places. We have writers, reviewers, webinar presenters, and the like. And we do appreciate very much all the work of our subject matter experts that makes our work possible. Next slide, please. If you are interested in joining CAST as a member, please do scan the QR code on your screen at this time. There's a lot of opportunities for you to engage with us, and those are listed on the screen. We'd love to have you join our ranks. Next slide, please. If you want to learn more about CAST, do indeed on the left side of your screen, click on that, scan that QR code or click on it, excuse me. And then on the lower right, you can reach out to me via LinkedIn. Chris Boomsma on LinkedIn there. That QR code will get you right to my profile. Happy to chat with you. With that, now I'm going to hand it off, and I am going to welcome to the screen at this time to join me, Deepika Chauhan. She serves as Director of Plant Transformation and Delivery Technology at Pairwise. She has expertise in plant biotechnology and has guided innovative crop improvement projects and helped turn scientific breakthroughs into practical solutions for agriculture. She also holds the position of SIVB Membership Chair. And with that, welcome, Deepika. Thanks for joining today.
Thank you, Chris. On behalf of Society for In Vitro Biology, we are pleased to welcome everyone to this joint venture of CAST and SIVB. Founded in 1946, SIVB is a professional society with a mission to foster the scientific and technical growth of its members, conduct biotechnology research, and to understand the biology at the cellular and development level, promote, expand, and support the scientific knowledge base for the benefit of all society. Our mission is accomplished through the Society's two world-class journals, an online newsletter, annual meetings, workshops, and through support of teaching initiatives in cooperation with educational institutions. Today's webinar supports this mission. It is a second installment of our continuing webinar program. The SIVB membership and education committees are working on additional webinars, so please keep an eye out for additional program this year. Next slide, please. If you are a member and haven't renewed your membership for 2026, or you are not an SIVB member but want to support our mission, we encourage you to join the SIVB at members.sivb.org. Our membership is calendar-based, so all membership begins this month. Some of the benefits of joining SIVB includes, but are not limited to, online access to our publications, discounted annual meeting registration rates, providing opportunities to share your research and network at these events, volunteer opportunities, and recognition through prestigious awards. Next slide, please. And speaking of meeting registration, SIVB will be holding our 2026 In Vitro Biology meeting at the Peppermill Resort and Spa in Reno, Nevada, from June 6th to 10th in 2026. The program looks to be an exceptional event with sessions on advancements in animal and plant technologies, natural and induced genetic variations, synthetic biology, and use of AI tools and techniques in research, and so much more. Please scan the QR code on the left for more details of the program or on the left to register. We are also thrilled to share that SIVB will be celebrating our 80th anniversary at the meeting with two special keynote presentations on Sunday from Dr. Indira Vasil and Delia Bethel, and a special reunion celebration on Monday, June 8th. I hope you will register to join us. Scan the QR code on the left of the slide or visit sivb.org/meetings for more information. Lastly, we would like to share the success we have achieved with our new postdoctoral mentoring program, MATCH, Mentoring and Talent Cultivation Hub, where postdoc members gained insights into trends and opportunities in plant and animal biology and biotechnology. They gained skills and knowledge from experts in the field to enhance their leadership and communication skills, and networked with individuals who shared common interest. We are filling positions for both mentors and mentees to participate in this year's program, and all participants need to be active members of SIVB. Please reach out to our SIVB business office at sivb@sivb.org or scan the QR code on your screen if you're interested in participating in this program. Now let's get started with the webinar. At this time, I would like to introduce my co-moderator, Dr. Ian Curtis, who will introduce our today's speaker. Dr. Curtis is a pioneer in plant transformation, founder of the SIVB webinar series, and co-founder of the Distinguished Service Award in 2014. Ian has been a member of SIVB since 2011. Since arriving in St. Louis in 2017, Ian has currently moved into consulting, advisory, director, and scientific roles with several startup biotechnology companies, including Benson Hill Biosystems, Plastomics, Edison Agro Sciences, Lone Wolf, and Upstream Biotechnology. Welcome, Ian.
Thank you, Deepika. Today's speaker is Daniel Jenkins. Mr. Jenkins has 30 years of experience in the food and agriculture industry, including prior roles as Managing Director and Chief of Staff of the Agricultural Section of the Biotechnology Innovation Organization in Washington, D.C., and leading Monsanto's regulatory team overseeing domestic chemistry and biotech crop approvals across the FDA, EPA, and USDA. He serves on the advisory board of North Carolina State University's Plant Science Initiatives Center of Excellence for Regulatory Science in Agriculture and is a board member of the American Seed Trade Association. He has published on gene editing policy in Nature Plants and in the Society for In Vitro Biology. He holds a Bachelor of Science in Biology from Cali Poly San Luis Obispo, a Master of Science in Entomology and Applied Ecology from the University of Delaware, and a Juris Doctor from Loyola Law School. After his presentation, we will offer the opportunity to address any questions you may have. You're welcome to place your questions in the chat during his presentation. The webinar is being recorded and will be made publicly available. Now, with introductions out of the way, it is my pleasure to welcome Daniel Jenkins. He'll be speaking on gene-edited crops and global regulatory navigation. Welcome, Dan.
Thank you. So I'll share my slides here. Give me just a moment. Okay. So thank you very much for the opportunity to speak with you here today. Thank you to CAST. Thank you to SIVB. It's wonderful to see in the comments in the chat people from all over the world and everybody telling each other what the weather is, which is kind of fun. I would say on behalf of North Carolina, it's also below average here and a little cold. So I'm looking forward to chatting with all of you today. So I wanted to start by telling you a little about Pairwise and our pipeline and regulatory experience, and who we are as a company. So Pairwise, what we do is we try to use this technology to make plants easier to grow and to eat in an effort to try to make the world a healthier place. And health has lots of dimensions. That could be environmental, it could be diet. And generally speaking, how we approach that mission is through partnership. And so as we move forward as a company, we like to say that the world is changing fast, but we help plants keep up. We believe that plants contain all the genetic information they need to adapt to our changing world, but it's up to us to deploy it. And so we see applications across all of these different areas: food security, human health, labor supply, supply chain durability, where this technology can really help. So some examples of some of the things that we have done, and again, these are through partnership. Um, I'll start in the upper left, where we've done editing in corn with Bayer. This is an example of where we were able to make some edits that could contribute to yield in maize very quickly. So that's one thing I want to emphasize here, which I'm sure many of you know, is that one of the biggest powers that this technology has is the speed at which you can get things done. So with that corn example in the upper left, something that we've done in row crops, we were aiming for a target that could increase yield. It turned out that doing a simple knockout turned that corn into a 16 row ear of corn into a 32, which we're not showing here because it looked like a soccer ball, which wasn't very useful. So then pivoting to a different approach of using base editing, we were able to make all of these different varieties with 16 rows or 18 or 20 or 22 all the way up to 32. And we can make all those varieties and then take them to the field and then test them and figure out which one gave you a yield increase without decreasing the weight of those kernels, the number that was in a row. So finding that sweet spot. And we did all of that in just a few years, which is pretty incredible. And the lower left is an example of doing editing for disease resistance, which obviously can help to reduce fungicide use. And the upper right, that is a picture of a cherry tree that we have achieved some proof of concept on. And that is a cherry tree that is flowering after just a few months, not a couple of years. So um, we see opportunity across different types of crops. And we've done some work that some of you may have heard about in mustard greens and sort of taking the mustard out of the mustard greens to make them more palatable, because underneath that is a nutrient dense, tasty base for salads. So we do this through our platform, which I won't spend much time on, but we've gotten pretty good at this. We've done over 11 million plant transformations to date in 15 different crops. And again, we're doing it across many different partners, whether that's through collaboration on some of the things I was just describing, like in corn or soy, or cherries, but also through licensing, including nonprofits and universities and institutions around the world. So I'd say that's probably enough about Pairwise. And what I wanted to do now is just tell you a little bit about the landscape that we all see here for policy. I know many of you are probably familiar with this already. So I want to give a little bit of that background and then walk you through with some examples of, I'm going to focus on blackberries for Pairwise today, and our experience in moving that through some of the regulatory systems. I'm going to particularly focus on Latin America, but with questions, I can go wherever you would like. But I'm going to start with the policy. And obviously, policy matters. And there is great importance to good policy because it facilitates um, lots of good things economically. So I've got a chart here that shows trade flows of agricultural products, exporters on the left, importers on the right. And obviously the economic aspects of agriculture are important. I don't think that it's lost on anyone that, and this is my view, that agriculture is probably the only thing that touches all of the UN Sustainable Development Goals. And so some of us wrote a paper here through the International Seed Federation about a call for action to foster good policy for plant breeding innovation because it can have such an impact and because it can facilitate such good things. So the question is, though, is what do we mean? What is good policy here? And what we're really focused on is pre-market regulatory policy. So this is those policies that you have to, or regulations that you'd have to walk through before you enter the market. We're not talking about things afterwards, like labeling or any of that stuff today, although I'm happy to answer questions on that. And there's lots of different approaches to pre-market regulatory policy for genome editing that could be applied. There's a focus on the process. Is that really what matters? Or is it the product at the end of the day? And Canada is a good example of a sort of strategy that really just focuses on the product. You could be classically risk-based, that we need to focus on the likelihood of harm. What is the hazard, if any, and what is our exposure to it? And then we'll need to adjust how you enter the market based on that analysis. There's hazard-based frameworks. It doesn't matter what the exposure is. There's just a potential to cause harm, and so maybe we need to ban something. There's the precautionary principle, which is based on uncertainty, aims to prevent the harm before it occurs. We've never had experience with this thing, so there's a presumption of a lack of safety that you've got to overcome, which can be quite difficult. Should benefit be considered at all? In the US, the statute that our Environmental Protection Agency operates under, called FIFRA, considers the benefit. Yes, pesticides kill things. That's a harm. It's sort of built in, but there's also this benefit that we need to consider that may outweigh that. There's also then at the end of the day, well, what is the form that some of these regulatory actions take? Can I put lots of submissions in a single batch? Can I do things early in R&D? Can I give you something hypothetical even so that I can get certainty on my path and begin to do research out in the field? So all of these things matter, and maybe on the right-hand side of the slide is a bit of a cartoon representation here about product-based tends to mean you'll treat these edited plants more like conventional, or do you start to move a little bit to the right? Is it conventional or conventional plus a couple of things? Or is it really getting closer to GMO, how you'll treat it, but maybe it's not as burdensome? Or do we just treat you straight up like a GMO? And you can see what the effects these different policy approaches have on market density or market concentration. So you can have a lot of players, like you see in conventional seed production for vegetables, for example, or in the GMO world, very high market concentration, and which is largely driven because of policy that makes it expensive and difficult to get a return on your investment. So which sort of innovation curve do you want to be on? What does this mean for investment and innovation when you think about these policies? And there's a fundamental challenge here globally with respect to gene edited products. It's a GMO mindset. And that isn't, the finger is not just being pointed at regulators here. It's true for us in industry, for many of us, that it's a big change in how we think about these things. And so part of my message, particularly when you think about small companies, but this is also true of big, but they wouldn't be as maybe not as badly affected by it given their size, is that even really light data requirements will lead to overwhelming a system when it comes to these genome edited plants or PBOs or PBIs or NGTs or whatever you may call them in your area of the world. Because if we're looking at data, for example, on um, what you edited your plant, do we need to generate data for each construct that led to a phenotype? Do we have to do it for every guide that we used to reach a target and edit it? Do we have to do that for every line within a variety, every crop, even though it might be the same kind of edit? So when you ask for example, maybe an off-target analysis, do I have to do that for every guide, for every gene, in every line, for every maybe genome if it's a polyploid? And if I've got early stage R&D and I've got many, many lines and I just want to get to the field, that adds up really fast. And some data in some places that's more GMO centric may ask you for like generational stability data. That's hard to do with a tree. So the GMO mindset also applies to, we're just thinking about row crops. We're just thinking about a single edit. We're just thinking about you coming to us at the very end. When you look at this paper, which I like quite a lot, that some folks at Bayer, I think, put out, that's the current state of thinking, that we have this maybe GMO, and then we do is we breathe that trait into all these different lines, and so they all sit underneath that umbrella. Versus if we have a good trait, then I'm not going to go through conventional breeding. I'm going to edit line 1, line 2, line 3, line 4, and they're all going to be identical with respect to that edit or highly similar. Do I need to generate that data every time? And this is again can lead to an overwhelming of the system very, very quickly. So this GMO mindset is something that we're, I think, doing a good job actually globally beginning to move past. But uh, we've all got to sort of get through this. So I have to talk about Europe here because this was such a big deal. And as I'm getting through sort of laying the landscape here, um, I'm sure many know about the European Court of Justice decision that said, when we look at this, NGTs, genome edited plants, yes, we're going to consider these GMOs. And that includes traditional mutagenesis, but said those are okay because they have a long safety record that sort of satisfies the precautionary principle. And this decision had a massive global impact that it sort of slowed innovation, created a lot of uncertainty, it slowed investment. And so EU right now, however, is really doing a great job, I think, of turning the ship around. The European Commission put out a proposal. They had to come to a three-party discussion with the Commission and Parliament and the EU Council. They just finished that and came up with a legislative proposal that now will be voted on by Parliament probably at the end of next month. And if that happens, and it seems more likely than not it will, then the EU will go through with regulations that allow for some of these products to be treated more like conventional and not as a GMO. So these new genomic techniques, or NGTs, there's a class here that would be called NGT1, which would be exempt from being called a GMO. There's some limits on it. There were some issues to work through. You can also be considered maybe an NGT2 if you don't qualify as NGT1. NGT1 really comes down to lack of foreign DNA, some limitation on the number of edits allowed, and there's a couple of traits like herbicide tolerance that won't be allowed. But that doesn't mean you're a GMO. You can maybe fall into this middle ground, which might be a little bit less burdensome. And so um, So this, we're hopeful that regulations could be in place by 2028. And I do want to point out here just one observation before I wrap up. What's the state of play with the globe? The basis for many, many regions and countries' regulations was based on the Cartagena Protocol and from the Convention on Biological Diversity. And this is the source of the precautionary principle. And it's interesting that many of the countries that took on this approach for their regulatory um, uh, constructs, that they basically said that when we focus on the fact that are regulations based on a precautionary principle, Cartagena Protocol, really focus on novel combination, that if we think that these kinds of edits could occur through conventional breeding, then they're not truly novel. And in some ways, that made it much easier for countries around the world that were very difficult from a GMO standpoint to make a policy change that allowed for a pretty low burden approach to genome editing. So with that, Argentina came out first in 2015 and with a really good policy. They allow you to go in early with hypotheticals. It fundamentally comes down to showing that you have a lack of a transgene, and they show their data on this often. They typically publish it. This is from the pictures from a conference I was at several months ago in Europe, where Argentina was showing the great diversity of developers working in different types of products and different types of traits. And so what you see as you think about that scale is because they've taken this policy, they're seeing a real flourishing of diversity in the marketplace, which is great. So that brings us to where are we in the world? So hopefully this works for everybody. This is a timeline from the International Seed Federation about policy developments as they've occurred from the advent of this technology for plants. And what you'll see really is Latin America really does lead the way here, and then it begins to spread. And Europe, you see red there with the European Court of Justice decision. New Zealand was an early red, and there's a lot underneath this. It's not quite as simple as this map would make it look, but generally speaking, green means things are pretty good. Europe is now trying to turn around, so it turns to a lighter green. And this is where we are as maybe about October. I just saw today that Vietnam is making some changes. Peru has put some things out. Expect some things from Panama soon. So things are progressing really well around the world. Okay, so our pipeline and regulatory experience. Um, and I'm seeing some questions in the chat, but maybe I'll get to those at the end. So I'm going to try to move through this with relative speed here. So our experience to date is we've moved through, we're getting close to 50 positive determinations that our gene edited products are equivalent to conventionally bred plants and not GMO. This is across 5 crops in 9 countries and about 30 gene targets. I think we probably have lots of others to thank for setting precedent that we could follow on, but in other instances, we're the ones that have set the precedent as well. And that upper right, those are fields of edited mustard greens grown in Arizona, and those are edited blackberries. And why berries? I like to say at the beginning of this talk usually that you probably don't care that much about blackberries, but I hope that you do by the end. Look, again, coming back to our mission, we're trying to make some of these things easier to grow and to eat, and in order to make the world a healthier place. And berries are difficult to grow. People don't like the seeds. And when we look at examples in the marketplace that have been really successful in getting people to eat more of a healthy thing, over and over again, you see some of the same characteristics. They tend to be available year round. They tend to be very snackable and convenient to eat. And if you get those things, then people will eat more of them, and that's a good thing. So blueberries are already snackable and convenient. A mutation was discovered. They don't have to go through a chill and a winter anymore. So they're grown in Peru and South Carolina and Florida now, and they're available all year. Baby carrots, very high profit margin here. They're 80% of retail carrots in the US. Snackable and convenient. These halos, these mandarin oranges in the US, they didn't capture half of the orange market. They captured half of well, they did capture half of the US market, but they increased total citrus consumption by 30% because they're snackable, they're convenient, they're seedless, they're easy to peel, and you can easily pack them in your kid's lunch. So blackberries, we see a similar opportunity. Highly nutritious. People like them, but they don't like the seeds. We can make them more snackable and more convenient. And what we don't want to do is invest a lot in trying to edit for nutrition or for flavor. These things are complicated. But what we can do is take an already great berry and make it better by improving the agronomics, by improving that experience by and tasting it with removal of the seeds. So what have we done? So one thing that we did is we've edited these plants to make them more compact. So they're smaller in stature. These plants are typically several feet tall and they take a while to grow. And if we make them smaller, we can do high density planting. So with high density planting, the rows are closer together and you can put more plants in a row. And so currently we have a field trial of these in Colombia as well as California. And so we can do up to about 3 times the density. We can get up to maybe a 50% yield increase or more. And it reduces, it's very reasonable to assume here that given that you're not putting all that effort into vegetative growth, that these are 4 feet, not 8 feet, that it really can reduce the amount of water or other inputs to produce a lot more berries. So we've taken that product and other products to places like Colombia, and I'm just going to focus on Colombia for now because Colombia has been wonderful to work with. And so Mexico is the largest producer of berries in the world. It is their second biggest export, but the regulations weren't in place. So we looked around and looked at different things to consider, like the growing climate, the regulations, IP protection, phytosanitary, et cetera. And we landed on Colombia. So the one thing is that Colombia did require you to have a physical presence in the country, which we didn't have. We're a small company to make a submission. Colombia ended up changing its regulations on that, which was great, made things a lot easier. We also had to work through phytosanitary and trade regulations. So it's interesting how some of these genome editing policies can force you into new places. And then you've got to go back and establish regulations for trade because they weren't there before. So in Colombia, these edited plants have been there for about a year now, at least. And what data was required for the submission? Well, this is a country who made an interpretation of the Cartagena Protocol and said they're not novel if you don't have foreign DNA in them. And so that's really the data that's required. Tell us what the edits are, um, and but show the data for the absence of foreign DNA. And the determination is that you're equivalent to conventional. Most of these have taken about 30 days, but we've gotten them done in a couple of weeks as well. We've done this in Colombia for both the mustard greens and several lines of blackberries. A single submission, you can put multiple plasmids and lines into it. And this is really representative of what we've done in other Latin American countries, whether that's Guatemala or Honduras or Argentina or Brazil, Ecuador. So, and like I said, this can sometimes lead to needing to work through new phytosanitary or market access issues. So these are the first edited blackberry trials in Colombia. So you can see in the upper left, these are the more compact ones. They flower sooner. They produce more. You can see the row spacing, they're tighter together, as well as more plants in a row. There's quite a sizable production of these and has been for quite a while now, like as I mentioned, in Colombia. And this again speaks to speed. So first we grew the unedited variety in Colombia in 2022, and 3 years later, the edited variety has been for sale and has been selling very well in Colombia since about September. So we did all that in 3 years, and that's very rapid. And we've also taken this approach. Those are all edited berries being grown in California. Of making sure that there's a QR code on the berries that we sell, because We believe in being transparent. We think it's an opportunity to talk about the benefits of the product. And so that we are sharing that these are edited, but again, our focus in communicating that is not only that this is a technology, but it's an opportunity to speak to what the impacts are that these kinds of products can bring to the world. And so as we go, as I mentioned, regulatory occurs really early with genome editing. You've got to get lots of plants in there, and we move through this commercialization process and multiple lines. So this is where we are today with the compact plants, but we're going to be moving into seedless varieties as well very soon. And the seedless, it still has a seed in it. It's more like a seedless grape where you just don't notice it there. It's very soft and it creates a totally different eating experience, which is really great. So these will follow on with the compact ones, and we'll continue to produce these in places like Colombia and Ecuador, and hopefully with policy change in Mexico in the future. Now, this is why I think you should really care about blackberries for a moment, if you don't care already, is we can take these traits and then apply them to other crops. So the idea here is that, well, we can take that cherry tree that I showed earlier, and we can make it pitless. And we can shrink that tree down, and in the upper right there, those are trees that are flowering and produce fruit in around 12 months and only grow to about 6, 7 feet tall. And so then you can cover tree production and you can mechanize harvest and you can do high density planting with something that hopefully takes a lot less resources and now lacks a pit. And you could do that to cherries and you could do that to peaches and you could do that to avocados and so on. And so that's what Pairwise is trying to do with products of our own. And I would just say here, this is my last slide, that this sometimes gets asked because I also oversee a lot of corporate communications. And we've done these announcements with licenses, for example, to Mars, who's using our technology and doing edits in cacao for cocoa supply, or our partnership with Sunworld on in the cherry area. And we monitor worldwide media uptake and sentiment. And what we've seen has been really surprising and very positive. The sentiment is almost entirely positive. It's been really very impressive, and these things have traveled around the world. So with that, I think I would summarize to say that good policy really fosters investment. That's what's happened in Colombia. And that's what's happened in Argentina and other places. And I look forward to it happening in places like Africa and Europe, et cetera. And the global trend is very much toward a low burden regulations that mainly focus on showing a lack of foreign DNA. We have to move away from this GMO mindset where these nice to ask and these additional sort of GMO like risk assessment questions just aren't necessary because they add up really quickly. And a smaller company will be less able to handle that than a big one. And you need to think about what can happen in terms of market concentration and whether that's what we want to do. The regulations have to be flexible in all early stage submissions with multiple traits across multiple lines, across a variety of crops. And with that, I would end and just say thank you.
Okay, thank you, Dan, for a
And I
fantastic.
see there's a lot of questions in the chat.
Sorry, thank you, Dan, for a fantastic lecture at this time. Are we on? Okay. Thank you, Dan, for a fantastic lecture at this time. We'll open the floor for questions.
I don't know if you want me to try to answer some questions or.
Reminder, please place your questions in the chat. I'd like to, as the instigator of this webinar, I'd like to put a quick question out. So in reference to the blackberry project, I have a horticultural background. So if you plant blackberries changing from 8 feet apart to 4 feet apart, are you not encouraging kind of increased like fungal diseases because you have less airflow?
So it's a good question. So far we haven't observed so much of those issues, and we're still tinkering a little bit with how dense do we need to be. But they're highly managed, but that could be a problem. You're right, but I don't think we've really seen too much trouble with that yet. But it's still a little bit early, so I don't want to say no. It could happen. Um, I'm seeing some of these questions like popping up here, and Ian, I don't know how much time we have, but
I'm going to fire away the next questions. So I know I've got Alan online here. So it says, I am regulated essentially appears to be straight lack of transgene policy. It appears that every letter whereby transgenes are no longer present in the final products come with the conclusion they are not regulated.
that's really close to correct. So ultimately, what USDA is really looking at, what they have authority for, is whether or not you have plant pest sequences that could rise to something that uh, might make your product a plant pest. And what that translates into is, do you have agrobacterium sequence or not, typically? And so, in theory, you could have transgenes in there that are not plant pest sequences, and they would also not be regulated. I would say that, but for all intents and purposes, that's pretty much right. I would note that there's been lots of submissions lately, including ones that included hundreds of genes uh, for, I think for maybe that was for soybean, and a single MI regulated submission, and that was fine.
Okay. So I've got a next question from Raj Sharma. Any work in other berries except blueberry, such as strawberry?
We've done a little work in other berries, particularly in something called black raspberries as well as red raspberries. We've done a little bit in some of these other berries that you mentioned, but we've been pretty focused on blackberry initially.
Next question. Okay, next question is from Stephen Walsh. Can you highlight examples in Africa, food, food security examples?
I think, but just from a purely pairwise standpoint, I think of the partnership that we have with IITA in from the CGIAR in Nigeria and the work that they're doing in yams, where they're using some similar approaches to modify the architecture there so that they're easier to grow, more productive for a staple crop. That's just one thing that comes to mind.
Okay. And another one from Stephen. Can you maybe highlight the regulatory challenges, opportunities in Africa for gene editing for food security?
I guess I would start by highlighting regulatory challenges. I think I'd start with some of the positives first. You've got places like Nigeria, Ghana, Kenya, others that have already set up really good systems and have already, I've just mentioned Nigeria in that example, they're already going. I would hope that South Africa would come around soon. I think that they probably will. My guess is that if Europe moves forward, then maybe South Africa will also. As terms of the specific crops for food security, I don't think I'm the best person. I would turn to people in Africa for that.
Okay. Next question is from Hannah Moshe. Curious if you're seeing any technology, including breeding software platforms or phenotyping tech that supports this regulatory burden. Seems like these technologies are primarily focused on product performance and informing selections.
I would say that we haven't had to use a lot of software platforms and technology for regulatory. I think that what we are doing is trying to use, um, AI and these kinds of things to help us with modeling certain types of approaches to edits that we think will increase the probability of success and make us more efficient.
Okay, next question from Fabricio Mendina, Bolivia. What are the labeling requirements for the gene edited crops like blackberries in Colombia?
They are considered like conventional, so the labeling requirements are the same as conventional crops. And so they're to be a little bit more pointed, there's not like a GMO labeling or anything like that. In fact, and that's the way it is in most places in the world for gene edited crops.
The next question is from Brad Doyle. Do you know of any edited soybean, corn, wheat, cotton, or peanut that are US grown and currently approved in the EU for export?
No, and if they had them right now, they would probably be waiting for the EU's new regulations to come into place rather than try to drive those through a GMO system. So there really isn't anything right now. It's still a little bit early days for all products um, to be entering the market, whether it's the EU or otherwise.
Next question from Stephen Strauss. Do you see any options for allowing inert transgenic DNA under non-GMO like tDNA borders or recombinant footprints?
Such a good question. So, um, and many of the countries that have whose policies flow from the Cartagena Protocol, I think that's going to be really tough. I do think that in a place like the US, there may be some room for that, and maybe that's a starting point that others could follow because, at least with USDA, that would not be of a concern from a plant pest standpoint.
Okay. Another question from Stephen. Can you maybe highlight the regulatory challenges, opportunities in Africa for gene editing for food security?
Oh, Ian, I think we did that one already.
All right. Isn't three before? Okay. Next one from Gary Brewer. Are Pairwise blackberries currently being marketed to growers and homeowners?
So the plants are being marketed to some growers now. That's really a very small group currently in California and in Colombia. And in Colombia, those berries have been sold to consumers.
Okay. Well, from Getu, no. Why do you need to check off target edits when you assess plant performances?
Good question. In most places, you don't. In some regulatory systems, they will ask you to check for that. And it's unclear what will happen in the EU with respect to that. Other systems may ask you a question that can be addressed with um, we design upfront to minimize that, and they might be okay with it. Others are going to ask you to do whole genome sequencing and look at off targets. It just depends. Generally speaking, though, it isn't asked for more often than not.
Okay, thank you. Next question is from Edwin Samir Barboza. Thank you for your excellent and very interesting presentation. Regarding the blackberry example in Colombia, were these varieties bred solely to reduce their size to increase planting density, or also to make them resistant to pest or disease? In which municipality were the trials conducted?
Yeah. So these were first we took this variety unedited and grew it in places around Medellin and Bogota, and other places. The unedited variety has been grown in Mexico. It's been grown in the U.S. for several years. We did not. All we did was this edit that modified the architecture and flowering time, and so any disease resistance is already there just as a part of the background of that variety.
Okay, thank you. So James Kober, what are your thoughts on gene-edited livestock? Same process?
James, that's a big question. So, I mean, I think that it's the same. It should be the same principles that if you could achieve it through conventional breeding, it should be treated that way. Like products should be treated in like ways. And but I know that what I showed with that global map, that was really about plants. Most governments still haven't really said what their policy and how they'll approach it is for animals or microbes.
Okay, next question. From Chetan Sonawana, hopefully I pronounced that correctly. What was the cost commercialization of hybrid developed through Cas9 genome editing technology?
Um, I really so we haven't done this through Cas9, by the way. We've done it through our own proprietary enzymes. What is the cost of commercialization? So that's a question really for berries, so which really aren't hybrids. I couldn't give you an estimate on that, but it's obviously in the tens of millions of dollars is what I would say right now.
Okay, thank you. Next question from Abiza Morien. Does gene editing technology apply to cocoa and coffee?
Yes, it applies to any plant you can think of, and specifically, we did this license to Mars, who is using it in cacao. With respect to coffee, I'm not aware of anybody using it there now, but I'm sure somebody is. Um, the thing I have to say here is that in the past, working in a tree crop wasn't really worth it for most tree crops because you'd take 100 years to get a return on investment. This is where the speed that gene editing can bring to a breeding program makes it worth it to actually do that. It's lowered the barriers.
Okay, thank you. So that question is from Mohammed Ahmed. Is there any gene edited rice variety approved in the USA for general cultivation?
I believe that there is, but I can't tell you off the top of my head, but I believe so. I mean, there's certainly, I believe there is. I just can't think of it right now off the top of my head, but you can go on the USDA website and find something there, I'm pretty sure. I'm also aware, as you may be, that there's several varieties that have begun to move through regulatory process in India, for example.
Okay, thank you. Next question is from Alfred. Well, regarding our delivery process, will the bombardment affect the regulation?
I'm not sure I understand the question. I don't know if that's about like how we're getting the enzymes in. Are we using like gene gun type of approaches? I'm not sure what that means. I'm sorry.
I think it is, yes. Next question, please. From Kyle Moeller. Can you please describe the EU's proposed NGT2 or GMO light framework?
I'll do my best, but Kyle, a lot of the details aren't quite there yet. So in theory, or maybe this would be an example, you've got a product that you have edited. Maybe it has over the 20 edits, the cap for NGT1, so it falls into NGT2, or you've edited it for herbicide tolerance, so it falls into NGT2. And then there's going to be a discussion of, well, what's the data requirements there? We don't really know. But I think that the point is that at the moment there's an opportunity to work through, and it may be on a case by case basis, what data is really required or not. Do you have to do all the animal feeding, for example? It's not quite clear yet.
Okay, next question, please. This is difficult to pronounce. I'll try. Basaprapalu Patil. Sometimes we don't see the edits on TO, but we can see them in T1 of tomato. Any explanation for this?
Oh, I'm sorry. This is like I'm not the right person to ask. I'm not sure why that would be.
Okay, thank you. Our next question is from Juan Luis Jurat Funantes. Do you foresee CRISPR knock-ins as a potential way to introduce insecticidal protein genes into crops instead of agrobacterium transformation?
Yes, and I think that um, disease resistance, insect resistance, it's going to be easier from a regulatory standpoint if you can argue that you're recreating something that's already in the gene pool in most instances, but you're going to need to do it without agrobacterium, or at least the agrobacterium can't be there in the final product if you want to be treated like conventional. I think the answer is yes there, and there's been a lot of progress on that front, as I'm sure many of you know.
Okay, thank you. Next question is from David Halifax. Do you see this as applicable to ornamental crops?
I think it's applicable to all crops. It just depends on whether or not that is worth the investment to do based on the product concept.
Okay, thank you. We have some more questions. Carl Moeller again. Can you please describe the EU's proposed NGT2 or GMO light framework?
And that's the one that we just did, Ian. So I think we've covered that.
Again, is that another one?
Yeah.
We're mixing it up a little bit. Okay, so we've got Alan Bank back on. What's his herbicide tolerance is as a marker, i.e. ALS co-edit, and not as a marketed trait?
I'm yeah, what if the HT is a marker? And so in other words, Ellen, I think you're probably getting at like what would that mean from a regulatory process? Well, in most areas of the world, it wouldn't matter so long as you show an absence of transgene. And there may be times when you are truly just using that as a marker. For example, if I'm making a banana tree that is edited for a certain property, I might need HT as part of my system as a selectable marker, but there's no intention to apply herbicide over the top of a bunch of banana trees. So in the EU, I think right now it's more likely than not that that would kick you out of NGT1, but maybe there's still some discussion there. So I'm not sure what it will mean. I'd hope that that would not mean, because it is only a marker, that you could not be NGT1, but we don't really know.
Okay, so I think we've got about 3 minutes left, so we'll try and follow some more questions. Adriana Castano, what's your technical opinion regarding the European idea to establish labeling for food LA derived from MBT's crops?
So the current legislation states that no labeling would be required for NGT1 crops, those crops that are treated like conventional. I think that's the right way to go. If they're like conventional, I would go back again and say like products should be treated in like ways.
Okay, do we still have time for more questions? No? Okay, thank you.
Thank you.
Thank you. Thank you, Dan and Ian, for sharing this important
And hi to
topic.
Dave Songstad and others.
Thank you to the cast for providing the expertise in supporting this webinar program, to the SIVB membership and education committees for planning and supporting this webinar series, and to all of you for joining us today. As a reminder, this event has been recorded and will be made available to online in next few weeks. SIVB will be holding our annual meeting and 80th anniversary celebration at the Peppermill Resort and Spa in Reno, Nevada from June 6th to 10th, 2026. We have an exciting scientific program with a phenomenal lineup of speakers, and we cannot wait to share it with you all. Learn more about the meeting at sivb.org/meetings or just by scanning the QR codes that you see on your screen. Thank you and have a wonderful day. I hope to see many of you at our next webinar in the spring of 2026 and in Reno for our 80th anniversary celebration next June.